everyone, and thanks for tuning into my seminar. I'm Kate Carbone, and I'm a postdoctoral fellow in the laboratories of Dr. Christine Moussion and Dr. Ira Mellman in the Cancer Immunology Department at Genentech in South San Francisco. So in this talk, I'll be sharing my work studying the spatial organization of the immune response in tumors and how it plays a pivotal role in determining whether a tumor will progress or become rejected. And in particular, I'd like to share with you our efforts to develop a novel preclinical model we call STAMP to study the tumor microenvironment, as well as some recent work on how the biology of dendritic cells shapes tumor immunotype and fate. So as an outline for the seminar, I'd like to present some background on how we think about cancer immunity in the big picture and what were the research questions that drove us to develop STAMP. I'll introduce the technology and go through some of the data we have already published and how that changed how we think about the immune response to cancer. And then I'd like to present my ongoing work implementing this model to look at the biology of dendritic cells. So by the end of this talk, my hope is that I'll have conveyed to you how we approach studying the antitumor immune response. Our work is guided by the overarching question: can we learn how to enhance T cell responses by studying their natural interactions within the tumor microenvironment, particularly with myeloid and antigen-presenting cells? And ultimately, our aim is to leverage these insights to improve therapeutic outcomes for cancer patients. So I've shared a few learning objectives listed on the page below. First, explain the significance of the cancer immunity cycle, outlining its 6 key steps and how they contribute to the body's ability to recognize and eliminate tumors. Second, identify the 3 T cell immunotypes and understand their relation to tumor regression or progression. And 3, describe one of the key cellular determinants of different immunotypes and their roles in shaping tumor fate. All right, so first the background. So how does immunotherapy fit into our arsenal of treatment options for cancer? We can broadly categorize most therapies into radiotherapy, surgery, chemotherapy, targeted therapy, and I'll group hormone therapy here too. And the newest weapon in our arsenal against cancer, immunotherapy. So most of these other approaches aim to directly poison or remove the cancer cells themselves. But with immunotherapy, we instead aim to bolster our immune system so it can do the work for us. And this is a fairly new idea in the history of cancer. So the first descriptions of cancer and the idea that cancers might sometimes spontaneously regress can be traced as far back as ancient Egypt, like 1000 to 3000 BCE. And the earliest philosophers of medicine, here Galen, hypothesized that there might be a connection between inflammation and cancer. So in the late 1800s, there were attempts to use bacteria to cause inflammation to fight cancer. But with the success of radiation and chemotherapy, this work was largely forgotten for almost a century. However, in the middle of the last century, we began to understand more about how the immune system actually works at a cellular and molecular level. And there were a number of breakthroughs around how we might actually develop treatments based on the biology of the immune system. So in the early 2000s, these treatments began to come to market with the first FDA approvals of immunotherapies in a number of drug classes. So cytokine therapy, cancer vaccines, oncolytic viruses, and of course, checkpoint inhibitors and cell therapies, which have come into wide use for a number of indications. However, as the use of these therapies has expanded, we've come to understand that there's a population of patients that do not respond well to immunotherapy, and that the particular nature of their tumors and the spatial organization of the tumor tissue, also referred to as the tumor microenvironment, can be a key barrier to the success of these approaches. So to begin thinking about the spatial organization of the immune response in tumors and how it can regulate whether a tumor will progress or become rejected, I'd like to start out with the cancer immunity cycle. So the cancer immunity cycle is a conceptual framework first proposed by scientists at Genentech about 10 years ago, and it encompasses much of what we understand about cancer immunology. So first, cancer immunology spans multiple tissues involving cells circulating between the tumor, the lymph node, and the blood. It's cyclic, which means that regulation at any step has the ability to become self-reinforcing or self-limiting. And this is a bit of a chicken and the egg conundrum, but let's say the cancer immunity cycle starts with antigen release at the tumor site, and that antigen is captured by dendritic cells and other antigen presenting cells. This antigen is then trafficked back to the lymph node where the antigen presenting cells can prime T cell responses. The T cells then traffic to the tumor site and infiltrate the tumor itself, where they're able to kill tumor cells. And then this cell death results in the release of more cancer antigens to continue the cycle. So, since the cancer immunity cycle was first proposed, we've come to understand each of these steps in much greater detail. But we've also come to understand that this original model of anti-tumor immunity overlooked one very important regulatory step, and that step is understanding the tumor immune microenvironment. So just last year, a revised version of the model was published, which more explicitly acknowledges that there are additional layers of regulation within the tumor microenvironment, which are essential for the process of antigen-dependent tumor cell killing. So factors shown here, such as the infiltration patterns of T cells within the microenvironment, their interactions with other cells, and their differentiation or proliferation can be very important for maintaining the effector functions of T cells in this very hostile environment. And so again, here it's depicted as a sub-cycle because just as with the primary cycle, local regulation can feed forward or become self-limiting. And this is the part of the cycle where my work falls. How can we learn how to enhance T cell responses by studying their natural interactions within the tumor microenvironment, particularly communication with myeloid and antigen-presenting cells? So the work I'll show today relates to the infiltration patterns of immune cells within the microenvironment. And we use the term immunotype, which refers to the frequency of occurrence or spatial configuration of immune cells and their functional states within or nearby a tumor. And different tumors can have distinct immunotypes based on the types, densities, and locations of immune cells present in the tumor microenvironment. So why is the biology of immunotypes important? Well, from a clinical perspective, it's been well established that patient tumors can be classified into 3 categories of spatial organization based on T cell infiltration. And we call these desert, excluded, and inflamed immunotypes. So if you take a closer look at each of these by IHC, you can see that desert tumors have basically a complete lack of CD8 T cell infiltration. In the immune-excluded tumors shown next, there are abundant CD8 T cells, but they're confined into the stromal bands of the tumor at the periphery and excluded from the actual tumor parenchyma or core itself. And in contrast to these first 2 immune-cold immunotypes, the immune-inflamed or hot tumors have abundant T cells and also robust T cell infiltration throughout the tumor parenchyma. And different indications have different frequencies of these immunotypes. So prostate cancer is canonically considered a desert, colon cancer is a more excluded indication, and melanoma most often exhibits the inflamed phenotypes. And these immunotypes have been shown to predict patient response to immunotherapy. So here is just one example, looking at immunotype and outcome in the context of triple-negative breast cancer. So you can see that patients with immune-inflamed phenotypes, and that's shown here in the purple, have better outcomes and better response to cancer immunotherapy compared to patients with excluded or ignored tumors, which ignored is another way to call a desert. So that's shown in the blue and the green below. So this leads to the obvious hypothesis that it might be therapeutically advantageous to drive an inflamed phenotype. And that brings us to the research questions which catalyzed the development of the STAMP model. First, can we find ways to boost T cell responses by analyzing their natural interactions within the tumor microenvironment? Or more specifically, we were interested in deciphering what are the drivers of inflamed, excluded, and desert immunotypes, because better understanding the characteristics and the triggers of these distinct immunotypes could provide valuable insights for their clinical management. Okay. So next I will take you through some discussion of the STAMP model and how it came to be and the ways that we're using it in our research. So when I joined Genentech in 2018, Christine Moussion was forming a team to devise a high-throughput technology that would allow us to study the biology of immunotypes in mice live and in real time. And we call this technology STAMP, for Skin Tumor Array by Microporation. And so this is our STAMP team, myself and another postdoc in the lab, Chimo Pechuan, as well as scientists Lupe Ortiz-Munoz, Marcus Brown, led by Christine and Ira, of course. And the power of STAMP for our questions is that it's a reliable way to generate tumors with all 3 different immunotypes, and it enables us to study these tumors in vivo using non-invasive microscopy, so we can watch tumors in real time and also track their fate over several weeks. So how does this system work? Well, the model is based on a novel application of an infrared laser device called the Pantech Please laser device. And this is a clinical device that was originally developed for dermatological and vaccination applications. And here we've repurposed it because the way it works is that it uses an infrared laser to create an array of pores in the skin of a mouse ear. And what we can do with these arrays is apply a solution of clonal tumor cells, then incubate with Matrigel. And when we come back about 4 to 7 days later, we can use non-invasive epifluorescence stereo microscopy to look at and monitor the growing tumors. So this is another image of how the system works. You can see an actual array of pores into which we're depositing these tumor cells for implantation. And so using the STAMP model, we can address our questions with a few advantages compared to other approaches. First, STAMP is very high throughput compared to traditional models. We have more tumors and using fewer mice. Next, because we have many tumors within an array that are populated by T cells that are all primed in the same lymph node, we can really focus in on the regulatory role of the microenvironment specifically. And so also, as I mentioned, STAMP imaging is non-invasive, so we can observe the full lifetime of a tumor, either progressing or becoming rejected, and then contrast those different outcomes. And finally, STAMP also enables high-resolution imaging, so we can directly measure immune cell biology in situ. So because it's non-invasive, this enables us to record both the tumor microenvironment in real time and also track the fate of individual lesions over several weeks. Okay. So this is an example of some of the data we can produce with the system. Here we've implanted an array of tumor cells from a pancreatic GEM model, or genetically engineered mouse model. And this is our workhorse cell line with STAMP. So it's a KRAS G12D, P16, P19 mutant cell line. And you can see that these tumors, which are shown in white, all sort of grow in an array. And remarkably, with this model, where we have fluorescently labeled T cells shown in red, we can identify immune inflamed lesions. You can see highlighted here, immune excluded lesions, and also immune desert lesions. And all of these lesions can coexist in neighboring tumors within the same array. And they share hallmarks with those clinical immunotypes that we described before. So as you can see in the inflamed lesions, we have T cells that are infiltrating throughout the tumor. In the excluded tumors, we have T cells confined to the periphery or the stromal bands. And in the desert tumors, there's negligible T cell recruitment at all. When we then look at the fates of these different lesions, what we observed is that the immune inflamed tumors have the highest rate of tumor rejection, just as we see in patients. So this here is a Kaplan-Meier curve, which is maybe a little bit reversed from how you might be used to seeing this type of data. So percent survival is shown on the x-axis, or is shown on the y-axis, and days is on the x-axis. But this refers to persistence of individual tumor lesions. So a decrease in survival means that a tumor is disappearing. So here in the immune inflamed condition, shown in green, you can see more tumors are becoming rejected, so the survival curve declines, which is ultimately better for the mouse. So everybody with me? I know it's a little counterintuitive, and I'll show a number of these plots throughout the talk, so I want to make sure we're all on the same page. Okay. So we wanted to dive into this a little bit more and understand how these different immunotypes were related to tumor outcomes specifically. And one of our first questions was whether this tumor rejection was in fact dependent on adaptive immune responses, as is known to be true in patients receiving immunotherapies. And so what we can see is that tumor rejection is dependent on T cells. So here we implant arrays in either wild type or RAG knockout mice, and we see that there is rejection of tumor lesions. These lesions here indicated in red disappear by day 14, shown in the second panel. However, in the RAG knockout context below, you can see that there's basically no tumors that are becoming rejected. And so again, if we look at these sort of, this is showing the individual tumor growth with tumors shown in red becoming rejected. And if we then look at these stamp Kaplan-Meier curves, we can see that there's really negligible rejection when we have adoptive transfer or in the RAG models, but when we have adoptive transfer of wild type T cells, now you see that survival curve declining. And what's more is that these T cell responses are antigen specific. So when we take the same RAG knockout model and compare adoptive transfer of wild type T cells versus adoptive transfer of OT1 T cells, and this is in a context where the tumor cell line does not express ovalbumin, so OT1 is the antigen non-specific T cells, we see that only in the wild type context are T cells robustly recruited to tumor lesions, and these T cells can mediate tumor rejection. Whereas the antigen mismatch T cells, here the OT1s, are not able to mediate tumor rejection. So this is showing, again, the growth curves. And if we look at this reversed Kaplan-Meier curve, there's only tumor rejection in the presence of the wild type T cells. So next we wanted to look at whether this recruitment of antigen-specific T cells is actually translated into increased tumor cell killing, and see if we could use the live imaging capability of STAMP to measure that T cell function directly. So to do that, we made use of a calcium flashing reporter system where we can express the protein GCAMP6 in tumor cells. And upon T cell cytolytic attack and pore formation, there's an influx of calcium into the tumor cells, resulting in an increase in fluorescence, indicating that there's active killing happening. So this is an example of the data here. And you can see that there's calcium flashing in a representative inflamed tumor and an excluded lesion. So here on the left, you can see in the standard deviation projection shown in the blue to orange heat map, there are these foci of sort of active GCAMP flashing, these sort of orange spots that are indicated. Whereas in the immune excluded tumor, there's much less of these hot spots of T cell killing. So this is what the data itself actually looks like in the inflamed tumor. And so you can see these calcium flashings, regions where T cells are actively killing the tumor. We can then quantify this data. So here, plotting the flashing index relative to the tumor growth change from day 8 to day 13. So you can see the inflamed tumors shown here in red have the highest flashing index, and they also are growing the slowest relative to the excluded tumors shown in blue. Okay, so far what I've shown you is that STAMP tumor arrays display the entire spectrum of immune phenotypes, that antigen-specific T cells drive an anti-tumor immune response, and that inflamed tumors display higher frequency of T cell attacks and lower tumor growth rates. So armed with this system, we wanted to look deeper into what are the cellular and molecular determinants of these immunotypes. We wanted to know, are these immunotypes stable or dynamic over time? And how does cancer immunotherapy impact immunotypes to promote tumor response? So the first question we set out to answer was whether the determinants of immune phenotype were tumor cell intrinsic or extrinsic. We wanted to know whether there were pre-existing differences in the tumor cell line or differences in the T cells that accounted for the different fates. And in fact, what we found is that inflamed, excluded, and desert tumors are genetically and transcriptionally identical at implantation and are populated by equivalent pools of T cells. And this was quite surprising to us. So I'll take you through the data here. First, looking at tumor cell heterogeneity. So this is an upset plot showing results from upset R plot showing results from whole exome sequencing, representing the genetic mutations present in tumor lesions of different phenotypes. And what you can see here is that even though this is a clonal tumor cell line, there do exist some genetic variants, 321 detected in total. However, the vast majority of mutations that are present can be identified in all three immunotypes. And when we manually curated the very few mutations that were not common across all of the immunotypes, we found only one mutation that was truly unique to the desert immunotype. And this variant was found only at very low frequency in those desert tumors. So from this, we concluded that the pre-existing genetic variants don't seem to be responsible for driving the different immunotypes, because the mutations that are present are not uniquely associated with only one single immunotype. They're common across all of them. So next, we wanted to look at the transcriptional variability of these populations. So to do that, we performed single-cell sequencing of our pre-implantation tumor cell line and identified 7 different transcriptionally distinct clusters. We then performed single-cell sequencing of our post-implantation tumor cells by biopsying individual microtumors, which we had classified by the different immunotypes. So we next mapped the pre-implantation clusters onto the cells from the post-implantation tumors. And what we found was that every transcriptional cluster we identified prior to tumor cell implantation was capable of giving rise to all 3 phenotypes, suggesting that these transcriptional differences were also not likely to be driving the difference in immunotype. So based on these genetic and transcriptional analyses, we ruled out that pre-existing tumor cell heterogeneity was the most important driver of immunotypes in our system. And so to clarify, we're not suggesting that the genetic and transcriptional context of a tumor cell line is incapable of regulating immunotype, but there's an additional layer of regulation at the level of the microenvironment. So here what I'm showing is 3 different cell lines we implanted with STAMP, a non-small cell lung cancer line, our workhorse KPP line, and the B16F10 melanoma. And you can see that these different cell lines have different propensities towards one immunotype or another. However, all 3 are capable of generating all 3 different immunotypes, suggesting that the regulation of immunotype at the level of a tumor microenvironment is separate from the transcriptional and genetic context of the cell line itself. Okay, so given these observations, we wanted to see what tumor cell extrinsic factors might be giving rise to the differences in immunotypes between lesions. And our initial hypothesis was that the discrepancies could be attributed to the T cells themselves. Perhaps different types of T cells would respond differently to the tumor microenvironment, and this would drive them to localize in different patterns. And so to test this, we performed single-cell RNA sequencing of T cells from lesions with inflamed or excluded immunotypes. Here, desert isn't shown because they don't have T cells. And what you can see is that proportionally, there's very similar representation of the different clusters of T cells. And so when we look at the 12 different subtypes of T cells, and we also classified the clonotypes. So here I'm showing the top 7 most abundant clonotypes. We can identify all of these types of T cells in both excluded and inflamed tumors, regardless of immunotype. So this suggested, again, that T cell heterogeneity is likely not the driving cause that is patterning the different immunotypes. Okay, so although I've shown you now that, mutationally and transcriptionally and in terms of their T cell infiltrate, these tumors are extremely similar, they are in fact different. So here, looking at a PCA plot from bulk RNA sequencing of the entire microenvironment, you can see that these tumors are in fact transcriptionally different from each other. And looking at gene expression in the different tumors, the excluded, inflamed, and desert tumors are in fact distinct, so they cluster separately. And importantly, they share features with their analogous clinical immunotypes. So if we look at the pathways that are upregulated in the different immunotypes in comparison to the ICON-7 phase 3 clinical trial in which ovarian cancer lesions were classified by immunotype, we can see that inflamed tumors in STAMP and in patients have high representation of these T cell associated pathways. So interferon alpha response, allograft rejection, interferon gamma response, and they're very low for pathways known to be associated with excluded tumors, including here TGF beta signaling. So we think that this is a really robust model for understanding these clinical immunotypes and specifically understanding the microenvironmental regulation that leads to these different states of tumors. Um, so I won't go into these experiments in great detail given the work is published, but we were able to identify a few key factors in the microenvironment that drive the immune inflamed phenotype. So in short, we generated a single cell sequencing atlas of the myeloid and stromal populations in the tumor microenvironment and noticed that there was a strong association of neutrophils and monocytes, shown here, and also a chemokine producing fibroblast population, but all associating with the immune inflamed immunotype. And we were able to perturb these populations using genetic models and antibody mediated depletion experiments to show that they play a functional role in determining immunotype. So these experiments led to our model shown here, whereby early in the tumor lifetime, there's a polarization of cancer-associated fibroblasts, here in yellow, towards a chemokine-producing population, which secretes a lot of CCL2, 7, and 8, which is then sensed by monocytes and neutrophils expressing CCR1 and 2. And in turn, the neutrophils and monocytes produce high amounts of the T cell chemoattractant CXCL9 and 10, thereby recruiting T cells in the microenvironment via the receptor CXCR3. So I encourage you to take a look at our publication to understand how we came to this conclusion. But in summary, for this section, we were able to get a handle on some of the cellular and molecular determinants of this immune inflamed immunotype. And next, we wanted to look at these questions of, are these immunotypes stable or dynamic over time? And how does cancer immunotherapy impact immunotypes to promote tumor response? So what we noticed in our data is these immunotypes are actually not stable. So this is an example of an individual tumor imaged at 6 different time points, and you can see that it's changing immunotype as we come back and look at it at different time points. And so here I'll also introduce a metric called the infiltration ratio, which is looking at basically the proportion of T cells present at the exterior or the stromal periphery of the tumor versus the interior of the tumor itself. So an infiltration ratio of 0.3 or less than 1 would indicate a more excluded phenotype, whereas an infiltration ratio of 1 means that there's sort of an even distribution of T cells throughout the tumor and the stroma. And we can represent this data in the form of a heat map. Oh, I apologize for the formatting here, but looking at every individual tumor, we can show individual tumors here on the y-axis and time on the days, and then color indicates the immunotype of that tumor. And so you can see that there's many different patterns or trajectories by which tumors traverse these different spatial states. And remarkably, when we looked at the survival of these different tumors, as I showed before, the immune inflamed phenotype is associated with the highest rates of tumor rejection. However, this is only true at the early time points of the tumor. So if we then come back and look at the last immunotype before rejection or animal sacrifice, now the immune inflamed state is not such a good predictor of the fate of that tumor. So we wanted to come up with a way to take advantage of the entire life history of these tumors to try to understand whether or not they would become rejected. So we can take these tumor immunotype histories and translate them into what we call these T cell ratio abundance plots. So now here, every point is a single tumor at a single time point, representing the total amount of T cells present on the y-axis and the inflammation ratio or the degree of T cell exclusion on the x-axis. And so as you can see here, the desert tumors have very low T cell abundance. An excluded tumor would have a low infiltration, a low inflammation ratio, but a high T cell abundance. And then the inflamed tumors are high inflammation ratio, high T cell abundance. And we can plot each individual tumor as it traverses this new state space. So when we then took all of these individual tumor trajectories or lifetimes of tumors and performed unsupervised hierarchical clustering, it showed that there were 3 key archetypical immune trajectories, and I'm showing those here. So in the first archetype, we have tumors that start out in a desert-like or lowly infiltrated state, which then recruit T cells immediately into a very excluded pattern. In archetype 2, as before, tumors begin with a very low infiltrated or desert type state. They recruit T cells into an excluded type pattern and then traverse into the upper right of the plot, or the more inflamed pattern. And there is also this population archetype 3, whereas before the T cells are not present, tumors begin in a desert type state. However, they immediately traverse into this excluded type pattern with high inflammation ratio, high T cell abundance. And so I've shown here the different states, so you can see how archetype 1, 2, and 3 progress. And when we then looked at the survival of tumors based on this full life history, we saw that this archetype 3 was the best predictor of tumor rejection. And in fact, the tumors that assumed this favorable trajectory were very highly rejected. Here, nearly 80% of tumors became rejected. So now I've shown you that, as we observed, immunotypes can be dynamic over time. And so we wanted to know, how does immunotherapy impact immunotypes or impact these life histories to promote tumor response? And so for this, we used a model of immunotherapy with combination anti-PD-L1 checkpoint blockade with anti-TGF-beta. And we saw that the combination treatment displayed efficacy with very high rates of tumor rejection. However, the monotherapies shown in yellow, orange, and the control in blue had very low rates of tumor rejection. And so what's happening in terms of the trajectories? Well, when we looked at this, we saw that each treatment was promoting a different type of trajectory. So in the control, we had our typical sort of trajectory, which looks a good bit like archetype 1, archetype 2. So here, tumors begin in a desert state, traverse to a more excluded state, and ultimately become inflamed. In general, in the control, there was relatively low T cell infiltration. And the pattern was quite similar with anti-TGF-beta. So again, we saw this archetype 2, or lowly T cell infiltrated state. In contrast with anti-PD-L1, we see that tumors actually very rapidly recruit T cells. However, it's into a more excluded pattern. So this is more of an archetype 1 type trajectory. And so all 3 of these first treatment groups don't show increased tumor rejection. And only the combination therapy, which recapitulates the archetype 3 trajectory we saw in our initial data set, where tumors become rapidly infiltrated with high T cell abundance, those tumors were the ones that became rejected. So to summarize this section, I've shown that tumor immunotypes are dynamic over time. Tumor survival probability depends on the time point of immunotyping. We've introduced these T cell ratio abundance trajectories, which allowed us to use the entire tumor immune history to predict fate, and shown that early and stable conversion towards the immune-inflamed immunotype is the strongest predictor of rejection at baseline and in the context of immunotherapy. And so just to kind of summarize here the importance of this work, I think at the conclusion of this project, we made 3 key contributions to the field. So first, our work challenged the idea that immunotype is a stable characteristic of a tumor written in stone in the mutational landscape. Both in that the microenvironment appears to play such an important role in patterning immunotype, and also that the immunotype of a tumor can change over time. And then second was the cellular circuit we defined to explain how the inflamed immunotype is programmed through this quite complex communication between fibroblasts, myeloid cells, and eventually T cells. And this mechanism really underscores that the interaction between a tumor cell and a T cell is not occurring in a vacuum. There's actually an intricate network of interactions between many different cell types that are influencing whether that primary interaction will in fact result in tumor rejection. All right. So with the time left, I'd like to share a little bit about the dendritic cell biology in this system. And so, as I presented in the beginning, we know that this immune-inflamed phenotype is the best predictor of, or is a good predictor of response to immunotherapy. However, as you can see here, not 100% of patients are responding to immunotherapy. So to illustrate this point another way, I've contrasted two different indications, desmoplastic melanoma and non-small cell lung cancer, which are canonically considered inflamed tumors. Both have very high tumor mutational burdens. And desmoplastic melanoma is induced by chronic UV exposure, whereas non-small cell lung cancer by exposure to carcinogens. So given our immunotype hypothesis, both tumors should respond extremely well to checkpoint blockade. However, in melanoma, we have an objective response rate of about 70%. And by contrast, in non-small lung cancer, there's a very low response rate of about only 20%. So what's different about an immune inflamed tumor that responds to therapy and one that does not? And what are the drivers of this immune inflamed phenotype overall? So we had a number of hints from the literature that dendritic cells might be important in supporting T cell function from within the tumor microenvironment and may underlie this difference. And this could be through a number of mechanisms, through chemokine production, inducing transcriptional and epigenetic changes in T cells, expansion of antigen-specific T cell clones, or transpresentation of cytokines and other survival and functional factors for T cells. So we had the hypothesis that dendritic cells may be important, and we wanted to know, are they important in differentiating responders and non-responders? And if so, how can we leverage the unique strengths of STAMP to gain new insight into how dendritic cells support tumor rejection? And we've seen that, in fact, dendritic cells, specifically CDC1 dendritic cells, promote tumor rejection in STAMP. Here I'm showing two different cell lines, B16F10 melanoma versus our workhorse KBP, in which dendritic cells are required for tumor rejection. And in dendritic cell deficient BATF3 knockout mice, we lose that spontaneous tumor rejection. And based on this result, we developed a purpose-built transgenic model for studying dendritic cell biology in STAMP. And so in this mouse model, we're able to look at dendritic cells labeled with TD tomato, T cells labeled with GFP, and our cancer cells expressing BFP. And this is an example of that data. Oh, I apologize, the movie's not showing. But through this, we can very precisely localize these different cell types to study their function in tumors. And so when we then classified tumors based on both their T cell immunotype and also their dendritic cell immunotype, we observed three primary categories. So our T cell inflamed subset, we could now divide into inflamed tumors that were infiltrated by dendritic cells and inflamed tumors that were excluded for dendritic cells. And we saw that the vast majority of our T cell excluded tumors were also excluded for dendritic cells. When we then looked more deeply into the survival of these tumors, we saw that that population of T cell inflamed tumors could be split by this additional layer of categorizing the dendritic cell immunotype, and having co-infiltration of both cell types together led to better tumor rejection. And using our calcium flashing model, we saw that there were higher rates of tumor cell killing by T cells, shown as these foci of high GCAMP standard deviation. And that's quantified here, showing that our co-infiltrated tumors had the highest rate of tumor cell killing. All right. So in this section, I've showed you we've developed a purpose-built transgenic model for dendritic cell biology in STAMP. We've shown that dendritic cells and T cells exhibit these doubly excluded, doubly inflamed, or the mismatch dendritic cell excluded, T cell infiltrated spatial patterns. And we've shown that these II or co-infiltrated type tumors with dendritic cells and T cells have more cytolytic attack and better tumor cell rejection. And so now our work is focused on really exploring what are the cellular and molecular hallmarks that are supporting better rejection in these co-infiltrated tumors, and how might we be able to leverage this biology to develop better ways to modulate the microenvironment to promote response. So to briefly leave you with an idea of some of our future objectives, we'd like to explore the dynamics of DC T-cell interactions within the tumor microenvironment over time to ask the question, when are these interactions actually required? We'd like to leverage the statistical power of STAMP, having so many lesions present in one array for spatial profiling to develop a molecular map of a rejection-associated tumor microenvironment. And last, we've been investigating the targeted manipulation of chemokines or other factors to shape the immune landscape within tumors to promote tumor rejection. And so I'll leave you with the technical advantages of the STAMP model again and how it has furthered our research. First, the high throughput nature with efficient data generation with more tumors and fewer mice. The ability to focus on regulation within the tumor microenvironment because shared lymph node priming enables that very precise measurement. It's a longitudinal approach where we can look at the full tumor history using non-invasive imaging, and we can pair that with high-resolution imaging to capture immune cell biology in situ. And so our goals are to start with first principles to advance strategies for immunotherapy. So looking at antigen-presenting cell T-cell communication to understand how antigen-presenting cells like dendritic cells support T-cell function within the tumor microenvironment, to understand these different layers of tumor cell intrinsic regulation that interface with the microenvironment regulation. How do those factors relate to each other to promote tumor rejection? And last, how can we take our understanding about the tumor microenvironment to engineer a better T cell for activity in solid tumors? And so with that, I'd like to thank my collaborators and colleagues, shown here as the Cancer Immunology Department at Genentech. And I'd like to call out a few specific people. First, the Mellman Lab and the Moussion Lab, who have all contributed greatly to this project, as well as people throughout the department, our support through core facilities and staff, as well as, of course, the STAMP team. So this work was definitely not done alone. Lupe Ortiz-Munoz, Marcus Brown, and Chimo Pechuan were really fabulous contributors to this project, and it was a joy to work with them. So with that, I'd like to thank you for your attention, and I hope you learned something new about how the tumor microenvironment regulates the response to cancer immunotherapy.